Yeah, I'm Zayn. This is Yusuf, Salman, and Hamza, and we're presenting C1.3.4 to C1.3.6 for the photosynthesis. Table of contents. First, we're gonna go over what's called the overview. Then we're gonna do 1.3.4, then 1.3.5, then 1.3.6. Overview. So, overview of photosynthesis. Photosynthesis is the process by which plants, algae, and some bacteria use sunlight to create oxygen. Food is the basis of food chain and provides most of the energy humans use. Process plants absorb water through their roots and carbon dioxide from the air, and then the water and the carbon dioxide are converted into oxygen and glucose in the plant's chloroplast. And the plant releases oxygen to the air and then stores glucose for the energy. This is basically the process of photosynthesis works.
Alright, so C1.3.4 is about the separation and identification of photosynthetic pigments by chromatography. So if you guys remember, we did chromatography in AP Chem. Basically, it's used to separate plant pigments. So in order for it to work is, you would first transfer the plant pigments to chromatography paper. It's like a special type of paper designed for chromatography specifically. And then you allow the paper to come in contact with a chromatography solvent. Usually it's like ethanol or methanol. And then after that, the pigments will be separated. They'll like move up like as you can see right here, they'll move up the chromatography paper. I know it's a little bit low quality. And then after that, you would measure the distance between the solvent traveled and each pigment traveled. So as you can see, each one here, so like chlorophyll B, chlorophyll A, xenophile, and I believe that one's Caravan, but yeah. Anyway, so this is an RF value, which basically measures the distance the pigment traveled over the distance of the solvent. Is it like a cut?
Do you want to zoom into that?
No, it's like cut a little bit. It's like supposed to be big. Yeah. Yeah.
Well, you can cut it out.
No, we could just edit it out. It doesn't matter.
We're just making the presentation.
Full screen.
They use that in crime scenes, actually.
The chromatography.
Yeah.
Yeah.
Yeah. So, the most common pigments identified by the color and RF value. So first we got chlorophyll A. It's basically like a little bluish green and has a really low RF value. And then we have chlorophyll B. It's like a yellowish green and it has a slightly higher RF than chlorophyll A. And then beta carotene is orange and has a very high RF value, like right here, this one. As you can see, it's like significantly higher than the rest. And then you have xanthophylls, which is yellow, and the RF value is between chlorophyll and beta carotene. So it's like right here, not as big of a difference, but chlorophyll A and B are like really close, and obviously carotene is like way bigger. But yeah.
He has a question.
Yeah.
Ibrahim Mohammed.
Yeah, what's it called? I was going to talk about that. So RF value, it's basically like a formula on finding what's called, basically, you know how like, yeah, exactly. So the RF formula, it's the pigments traveled on the chlorophyll paper over the solvents traveled on the chlorophyll paper. If you want to go back, and then because it says
It'll just help you know what
right
kind
here.
of pigment
So the
it is.
RF value is distant pigment traveled over distant solvent traveled.
It's like how long it stays on the paper.
Exactly. So what's called, when you calculate that, you get the RF value.
Yeah. And if you guys-
This is important. You should write it down.
Yeah.
rather than in your book.
Yeah, so I'll keep it on this slide if you guys want to take notes.
If you want to
If
take
you guys remember, like AP Chem, how we got like a little cup and then we put the solvent in it, and then we got a toothpick and we attach it to chromatography paper, and then we put 3 pigments and then watch them go up. That's basically what chromatography is. So yeah, you can do that experiment at home. Actually, just be very careful, don't do that at home.
You guys done writing the notes? Yeah.
You guys good?
We'll give you guys like 60 seconds to write this slide down.
Just by the way, pigments are the part on the leaf that would typically absorb the sunlight.
So we're gonna talk about 1.3.5, which is the absorption of specific wavelength of light by photosynthetic pigments. So first we have there's light spectrum and there's the next one is the pigment molecules. But first we're gonna talk about the light spectrum. So light electromagnetic spectrum includes all the forms of energy that travel in waves. So here we have different types. There's like radioactive sources that have like very low and more energy in the shorter lengths, and there's the higher, they have less energy. So basically there's in between the big ones and the small, there's a visible spectrum, which is approximately about 400 to 700, 800 nanometers. It's a portion of sunlight used in photosynthesis. So basically here it goes in order from the highest to the lowest. It goes from red, orange, yellow, green, and then it goes here blue, indigo, and violet, which is basically a Roy G bill, if you want to memorize it. So this is the center, this is like the basic numbers, and then it goes into positive and negative, and it's really big numbers. The second one we have the. You want to write it down?
The only important part about this is basically the 400, 700 nanometers, because anything that falls out of those values really shouldn't like concern you. I mean, they should concern you as a human being, but you can't like see them, so it doesn't really matter that much.
Yeah. So like you see, there's X-rays, there's AM radios, microwaves, all that. Only the visible light is the one that's visible. The second one is the pigment molecules. They're basically like chlorophyll A, B, and carotenoid. They absorb light energy. So basically, the first one, chlorophyll A, it's a primary pigment that absorbs the red and blue light. So the first one, the A, you got this blue and the red. Secondly is the B and the carotenoids. It's accessory pigments and broaden the range of absorption. So this is kind of a pigment molecule right here. It's separated into two. So this one right here in the pink is the membrane anchoring region, and this is the light absorbing region, which got the red and the blue. And how it works is that each pigment has electrons. It was kind of covered in the last slides. And they at specific energy levels, they the light excites the electrons radiating to a higher energy state, like what Ibrahim was talking about last slide, last presentation. And also the excited electrons are delocalized, that means they move freely, which allows them to energy transfer. And lastly, the energy utilization is that the energy the excited electrons is used to generate ATP and NADPH. So this is what happens with the pigment molecules. It creates the energy and it gets the ATP during the light-dependent reaction. Yeah, this is what Ibrahim was talking about. And the power of the Calvin cycle is where the chemical energy is stored in glucose.
Alright, now we're going to talk about C1.3.6, our last section. This is similarities and differences of absorption and the action spectrum. So here you're going to see two different graphs. Now, before we jump into this, I want to explain how we actually see things. So let's take that green chair for an example. That green chair is getting light from up there. That's the light source. It goes at the chair and then bounces off. It bounces off and then comes to our eyes, right? So that chair is really good at absorbing every color but green. It can't absorb the color green. That's why green is reflected and we perceive it as green. So moving here, pigments basically do the same thing. So we're going to take an example, I believe, in both these graphs. So chlorophyll A and chlorophyll B, I believe this chlorophyll A. You can see, this is the, alright, let's start with this one. This is the rate of absorbance. This means that on this scale of color, like we said, from 400 to 700 nanometers, this is our scale of visible light. This measures absorption. So this goes up here. So at, let's say, blue, it can absorb it really well. And at around like a red color, chlorophyll B can absorb that color really well. But then you look over here, in like the greenish yellow area, it can't absorb it. So what does that mean? It means it reflects the light and we perceive it as green. So basically, what I'm trying to get at, and the same thing for this one too. But basically, what I'm trying to get at is different pigments will absorb and reflect different wavelengths of light at different they'll just do it differently, yeah, at different rates, and like they'll appear to be in different colors, which means that they will use certain wavelengths of light better than others, if you know what I mean. This one, however, measures the rate of action / photosynthesis. So like we said, this one's chlorophyll A, so we'll focus on chlorophyll A. It can absorb green light. It bounces it off, so that's why we perceive it as green. Here, on the y-axis this time, it's the rate of photosynthesis. This means that, and it makes sense if you think about it, it's really high when it's around like blue, purple, just like it is here. That means that the light that it absorbs and doesn't reflect will be used in the process of photosynthesis. So we have white light, it separates it into this, and then it takes the parts that it can use. So the light, the green part would be reflected off like that chair. It would be reflected off and we perceive it as green. Otherwise, the other portions, which would appear as red, for example, in this case, or purple or blue, would be used by the pigment for photosynthesis. And basically, yeah, they're just two graphs with the same like flow, but they just mean different things.
Hold on. Bring that to place.
So are you saying that like the pigments, they use the energy from the pigments in order for the process of photosynthesis? Like
So
those? Yeah.
basically, these two graphs they measure two different things. Absorption basically measures light. This graph, what's called for action, it measures for photosynthesis. So basically these two graphs are really important. You guys want both of these in your notebook. But this one here, it measures what happens after the absorption of photosynthesis, and then like the wavelengths matter because they're like transmitted into the plant, and it's used for different things. Here it just tells you how some things reflect, like how they reflect light.
So it's
That's
like
the absorption, and then this
which
is the
wavelengths
action.
are going to give you the best rate of
Exactly.
Yeah, it's kind of like the one I explained, from 400 to 700. Those are visible, so the color, it has different color.
The color is just
the
what's
colors,
reflected
and
and
they're
not
reflect,
used
yeah.
for
Depending,
photosynthesis.
also the wave, the higher the wave.
And the two major differences between the graphs is one measures photosynthesis, and then the other measures absorption.
Yeah, I just want to give an example. This is chlorophyll. It's a picture of chlorophyll A. It's green. It reflects the green light, so it doesn't use the green light in creating the in photosynthesis. Instead, it will use like the purple or red light, which would be at like 400, 700. And then same thing. It'll absorb it and it'll reflect it and look green. Now, the main point of this lesson actually is about the similarities and differences of these graphs. So I just wanted to start off with the similarities. They both start at the 400 nanometer mark, violet, and end at the 700 nanometer mark, because that's the range of wavelengths that, like Samir said, is perceivable light to us. The x-axis will always represent the different wavelengths of light that can come into contact with the pigment. And in both the action and absorption graphs, so both of them, the lines will connect at the major troughs and peaks, meaning that they will flow in the same general trend or curvature. So if you see here, like this one correlates with this correlates with this correlates with that correlates with that. So it all flows together, even if it's
using the absorbed energy for photosynthesis.
Now, contrasting them, on the action spectrum specifically, it employs the y-axis as a percentage of the maximum rate of photosynthesis that can occur within a range of 0 to 100. So it would basically give you the percent. So if it told you that it was 80 at like violet, or purple light, it means that 80% of the maximum photosynthesis rate will happen when you have purple light. And then the absorption spectrum, it uses the y-axis as a percentage of the total light absorbed in arbitrary or with arbitrary units. But this is mainly what we need to focus on. It's a percentage of the total light that's being absorbed. That's it. This one I'll just skim over it. It's just how to measure the rate of photosynthesis. We can use oxygen production. So for example, an aquatic plant, we could submerge it, and then if we apply sunlight, oxygen will like come out, and basically like we can measure how much comes out. CO2 production is also how we can measure the rate of photosynthesis. We had a blanket, but
If we don't have time to, you guys could do it on your own for review and stuff like that, so you guys could answer the questions. Because sister wants you guys to answer the questions on the doc in your notebook, so if you want to. Or Nada has a good idea, so we can do it next lesson.
Can we do the science